Welcome back to Antibody Basics by Biointron, Part 3. In this episode, we'll cover bispecific antibodies and other multispecific formats. Engineered antibodies are modified versions of antibodies that have been designed to improve certain properties or functionalities, such as enhancing their therapeutic or diagnostic potential. Monoclonal antibodies have two identical binding sites which each recognize the same target antigen. Bispecific antibodies have two unique binding sites directed at two different antigens or two different epitopes on the same antigen. The first bispecific antibodies were described in the 1960s when antigen binding fragments from two different polyclonal sera were reassociated into bispecific molecules after reduction and reoxidation. In the 1980s, the first monoclonal antibodies were generated through hybridoma technology. Later on, the first article demonstrating the therapeutic potential of bispecific antibodies was published in 1992. During the 2000s, the first approvals of bispecific antibodies for therapeutic use took place. And now, in the present day, ongoing advancements in antibody engineering and further approvals of bispecific antibodies are occurring. Why choose bispecific antibodies? Multispecific antibodies can bind to more than one target simultaneously, thus resulting in higher binding specificity. They are advantageous over monoclonal antibodies in clinical therapeutic effects, as improved function translates into improved response rates. This is especially relevant for cancer, as direct specific effectors of the immune system can target tumor cells, enhancing cytotoxicity. In addition, you can optimize costs by this being an alternative to combination therapy with two monospecific drugs. The format of a bispecific antibody will depend on the research purpose and aim, with the two main categories being the single-chain-based format and the IgG-based format. The IgG-based format can be further categorized into asymmetric and homodimerized formats. Presence of Fc regions, symmetry, specificity, and number of binding sites can be very different between bispecific antibodies. Their small size allows them to be used to link immune cells to tumor cells, creating an immunological synapse. They are rapidly cleared from the blood within minutes and have better diffusion into tumor tissue. They also have higher potency due to closer proximity of interactions in the two paratopes and have ease of large-scale production in microbial systems, in addition to less immune-related adverse effects due to the lack of Fc. The second type of format is IgG-based. They have an extended blood half-life of about a few weeks due to recycling by neonatal Fc receptor. They possess their own crystalline fragment-mediated functions. They have a larger than single-chain-based format and a longer glomerular preservation. They also have improved solubility and stability, and triggers cytotoxic and T-cell priming effects, as well as being able to be purified by affinity chromatography workflows. While there are many bispecific antibody formats, here we have highlighted three of the most common ones. Knobs into holes involves replacing a smaller amino acid with a larger amino acid in the CH3 region of an antibody chain to form knobs. Meanwhile, one would replace a larger amino acid in the other chain with a smaller amino acid to form holes. A second format are the dual variable domain IgGs. This is a symmetrical molecular platform with 4 antigen binding sites and 1 pair of Fab domains. Each antibody arm utilizes short peptides to connect the two variable regions. The third type are bispecific T-cell engagers. This involves linking the CD3 antibody single-chain variable fragment and the tumor-associated antigen to redirect T cells to cancer cells for more targeted killing. Bispecific antibodies are particularly useful as therapeutics in cancer. There are several reasons for this, including the redirecting of immune effector cells to the proximity of tumor cells. They target tumor angiogenesis. They can have dual targeting of different disease mediators. They can block different signaling pathways simultaneously, as well as blocking cytokines, and being delivery vehicles for payloads to targeted sites. Many bispecific antibodies aim to treat cancer, but there are more starting to target chronic inflammatory, autoimmune, and neurodegenerative diseases, as well as vascular, ocular, and hematologic disorders. There are three main production methods for bispecific antibodies. The first common type is quadroma technology. This relies on the somatic fusion of two different hybridoma cell lines. This random pairing of heavy and light chains creates bispecific antibodies. Bispecifics produced through this way are similar to conventional antibodies. Catumaximab is the first approved bispecific antibody and was produced via a rat-mouse quadroma cell line. A second production method is chemical conjugation. Site-specific chemical conjugation involves lysine or cysteine-based cross-linkers. Alternative methods include disulfide bridging and incorporation of genetically encoded unnatural amino acids. Another type of production are genetic approaches. This utilizes recombinant DNA technology. Genes encoding the variable domains of two different monoclonal antibodies are cloned to produce a single construct. This results in the creation of bispecific formats such as scFv, diabody, or Fab. There are currently 14 approved bispecific antibody therapeutics worldwide and over 100 in clinical trials. The first to be approved was catumaximab. Approved by the EU in 2009, it was unfortunately withdrawn in 2017. It was aimed to treat malignant ascites. Blinatumomab was approved by the US in 2014 and is a bispecific antibody that treats acute lymphoblastic leukemia. Ozoralizumab was approved in Japan in 2022 to treat rheumatoid arthritis. Elranatamab got accelerated approval by the US last year to treat multiple myeloma. There are a wide range of potential applications for bispecific antibodies. One of them is to help with crossing the blood-brain barrier. Therapeutic antibodies may be re-engineered as a blood-brain barrier penetrating bispecific antibody. This would mean that one arm would be the therapeutic antibody while the other would be the transporting antibody. The transporting antibody would target an exofacial epitope on a blood-brain barrier receptor, thus allowing for a receptor-mediated transcytosis across the blood-brain barrier. Another application could be in diagnostic assays. In an immunoassay, a monospecific capture antibody can be immobilized onto a solid surface to bind to a corresponding antigen in serum. Since bispecific antibodies can function as a cross-linker, they can then be added to bind to the captured antigen and reporter molecule. Another application could be for the treatment of pathogens, specifically antimicrobial resistance, also known as AMR. AMR occurs when pathogenic strains are no longer affected by multiple antibiotics or chemotherapeutic agents. Recently, researchers developed a new bispecific antibody that targets the bacterial pathogen, Pseudomonas aeruginosa. The bispecific antibodies conferred 3 mechanisms of action and demonstrated the valuable potential of using multispecific antibody formats for the construction of therapeutic drugs. Developing therapeutic drugs requires processes across antibody discovery, expression, and optimization. At Biointron, we can take your gene sequence to purified antibody in only 2 weeks. Biointron's high-throughput platform enables the rapid expression of recombinant antibodies in HGK293 or CHO cells. We guarantee the delivery of 100 micrograms to 100 grams of recombinant antibodies. Remember, no antibody means no cost. We are also a well-recognized leader in VHH antibody discovery and production. With our self-owned alpaca breeding farm, we offer a 1 alpaca for 1 project commitment and guarantee the delivery of at least 20 unique binders, high diversity, and large capacity. We also have several other services, from bispecific antibody production to afucosylated antibody production and antibody optimizations. Thank you for watching, and comment with any questions or topics you'd like us to cover next. Don't forget to like and subscribe for more.